Our second step of gene expression is translation. In translation, the information found in our copied recipe, the RNA molecule, is read, and those instructions are used to make a protein. Again, using the kitchen analogy, this would be like making a meal. You can't eat the recipe, but when you make your meal, you can eat that. That is useful for you. Proteins themselves are very different molecules, just like a recipe and a recipe's copy are very different from the meal itself. The recipe is just the information, whereas the protein itself is what is useful for the cell. Proteins are not made of nucleotides like DNA and RNA are. Proteins are made of amino acids. The amino acids that you see here all have at their basic core this structure. The basic core structure of an amino acid is a central carbon atom attached to a carboxyl group, or COOH, an amino group, or NH2, a single hydrogen atom, and then a variable side chain. The variable side chain is what makes the different amino acids different from each other. There are 20 different amino acids, and so there are 20 different types of amino acids that can be incorporated into a protein. These variable side groups are pictured here as R, and you can see that different amino acids attach to each other between the carboxyl group of the first amino acid and the amino group of the next. Here, each one of these bonds is a covalent bond, and since it's a covalent bond between amino acids in a protein, that covalent bond between amino acids in a protein is called a peptide bond. Peptide is a term commonly used to describe proteins. So you're essentially saying it's a protein bond. This will continue, and you'll have proteins that are hundreds and sometimes even thousands of amino acids long. This will form lots of different proteins for the cell. Proteins are the major function molecule of the cell, and anytime the cell needs to do anything, it's going to need to make proteins to do it. So if the cell wants to eat or bring in food, if it wants to remove waste from the cell, if it wants to move or swim from one place to another, if it wants to go through cell division and divide, or make more RNA, protein, or DNA molecules, then it's going to need proteins to do that. Proteins, importantly, are almost always what enzymes are comprised of. So enzymes are almost always proteins, and enzymes help reactions go faster in the cell. Without enzymes, cells would die. And that's because the reactions would go too slow for the living system to sustain itself. So enzymes are extremely important in cells, and enzymes are almost always proteins. Another molecule that we're going to be talking a lot about this semester later on are antibodies. We've already started talking about them a little bit, but antibodies are proteins that your body makes to fight off disease, bacteria, viruses, and others. So as you can see, proteins are extremely important in cells, and in order for the cell to do anything, it needs to make proteins. Because these are so important, protein production is constantly occurring in all active cells on the planet. And that process is gene expression, the second step of which is translation. Translation itself involves lots of different molecules. So we've already seen here that translation involves a single RNA molecule that we'll now refer to as messenger RNA or mRNA, and that is the copy of the recipe that was made from transcription. In addition to that, you're going to need the ribosome. The ribosome is pictured here in this pink blob, and the ribosome is an enzymatic complex very large enzyme that is responsible for the production of proteins. The ribosome reads the information on messenger RNA and, with the help of another RNA molecule, tRNA, is able to synthesize and generate a chain of amino acids, which is essentially your protein. These tRNA molecules are very important in this entire process, so let's take a look at them and their structure. Your book shows the secondary and tertiary structure of tRNA molecules, but I'm just going to go over the tertiary structure because this is what it looks like inside your cells. tRNA molecules sort of look like an upside down capital L or a backward 7. There are two really important parts of the tRNA molecule. At the top here of the tRNA molecule, this is where the tRNA is attached to a single amino acid molecule. We can see that again in this picture earlier on in the chapter, where the tRNAs are pictured here, and those tRNAs are attached to a single amino acid molecule. This is what allows for the synthesis of proteins, is that the tRNAs bring over one amino acid at a time to form the protein. Remember, proteins are just chains of amino acids, and they're synthesized, much like DNA and RNA, one molecule at a time. How does the tRNA molecule know which amino acid to transfer? Well, that is determined by the bottom part here, known as the anticodon. The anticodon is called the anticodon because it is a complement to the codon found in messenger or mRNA. Again, going back to this picture here, we can see that messenger RNA carries nucleotide information as codons. A codon is a group of 3 nucleotides found in messenger RNA. So there's a codon here, and so forth. It's just a group of 3 nucleotides in a row in messenger RNA. The bottom part of the tRNA molecule contains the anticodon. The anticodon is pictured here, and here, and here. And as you can see, they match up or pair up, complementing the codon in the messenger RNA. Because this is RNA, G pairs with C, and A pairs with U, or uracil. So, how the tRNA knows which amino acid to transfer is it reads the codon from the messenger RNA by pairing up its anticodon to the codon of messenger RNA. So the anticodon is found in tRNA, and that is what determines which amino acid is transferred to the growing protein. Now, one of the things that I find amazing is that the pairing of the anticodon and the codon to determine which amino acid should be used is exactly the same in every cell on Earth. This is what we refer to as the universal genetic code. Here you actually have the information on how to make a protein. These are the codons, and this is the genetic code decoder. So when there's a codon that's made of UUU in mRNA, that is always translated in all cells on Earth as phenylalanine. UCU is translated always in all cells on Earth as serine, and so forth. This genetic code, as I mentioned, is nearly universal. And because it's universal, you can transfer a gene from one organism to another, and they will make the same protein. This is evidence that all life descended from a common ancestor, because the language of this genetic code has not changed in any cell on Earth. There is no reason for all this to be exactly the same in all cells on Earth unless all cells came from a common ancestor. Another common feature that you see in this universal genetic code is that it is pretty common for there to be more than one codon for a single amino acid. So you can have GUU translate to valine, but GUC is also valine, GUA is valine, and so on. Some amino acids are coded by 4 codons, others are coded by 6 codons, and a few are coded just by 2. This shows you that the genetic code is redundant. And what that means again is that each amino acid can be coded by more than one codon. This redundancy is helpful when cells go through mutations, which they can do on a regular basis. Even your cells are accumulating mutations throughout your lifetime. But often, this won't result in any negative consequence, partly because if GUU is mutated in your DNA to GUC, it'll still translate to valine, and there will be no change in the protein structure or function. So you can have a unique genetic code or a unique genetic sequence in your body, and it'll still make a functioning protein. This is one of the ways that you differ from other people in society, and one of the reasons why your DNA is unique to you is because you can have a functioning cell with all these mutations or differences. So how does translation work? Well, going back to this image, the messenger RNA attaches to ribosomes, either in the cytoplasm of bacterial cells or eukaryotic cells, or only in eukaryotic cells attached to an organelle called the rough ER. Then, when the ribosome attaches, it looks for what's called the start codon. Going back to our genetic sequence, the start codon in every cell on Earth is always AUG. So the ribosome looks for the start codon, which is the first AUG it can find in the mRNA, which here is pictured at the very beginning of the mRNA molecule. Often there are other nucleotides before here. Once the ribosome finds the start codon, AUG, it then starts to translate that amino acid sequence. So it moves to the next codon, UCC, which translates to serine, the next codon, GCC, which translates to alanine, and so on. And it'll move down the mRNA molecule 3 nucleotides at a time, or 1 codon at a time. And in doing so, we'll know which amino acid to place in the extending chain. This process continues until the ribosome hits one of the stop codons. Now, unlike the start codon, of which there's only one for every cell on Earth for the last 4 billion years, there are in fact 3 different stop codons: UAA, UAG, and UGA. These stop codons do not translate to any amino acid, but instead cause the ribosome to fall off of the mRNA molecule and release the newly formed protein. So we have the origin of replication that starts DNA replication, the promoter that starts transcription, and the start codon that starts translation. And likewise, DNA replication just continues until it's done, whereas transcription ends at the termination sequence, but translation ends at the stop codon. Make sure you know these for the quiz. In the next section, we'll talk about bacterial genetics.